Hi, Dr. Chernow here. So this is the third installment on the central nervous system, and we're going to discuss presynaptic and postsynaptic neurons. So this happens inside of the central nervous system. When we look at parasympathetic nervous system and sympathetic nervous system, we started within the central nervous system, within the brain or the spinal cord, or within here, right, brain and the spinal cord, and then we exit that into a periphery outside of the central nervous system. But now we're only inside of the central nervous system, in the brain or in the spinal cord. So this is a presynaptic neuron, and this is a postsynaptic neuron. As far as the target organ in the central nervous system, there's no such a target organ, because what happens, you have one neuron connected to another neuron, connected to another neuron, connected to another neuron, connected to another neuron, connected to another neuron. It's a spider web. Here, preganglionic. Ganglion is the collection of cell bodies, collection of the cell bodies that can communicate one with another. So here's the ganglion, here's the neuron entering, and within the ganglion you have the collection of cell bodies, like strings in the guitar. Here we're creating synapses between each neuron. We can stimulate postsynaptic neuron, or we can inhibit postsynaptic neuron. So for example, if this is the excitatory neurotransmitters being released, right, it should excite this neuron. And then we pass this information further down. But what if we inhibit this neuron? That all these neurons that in the series, in this chain, they will not fire. They will not transduct or conduct electrical impulse further down. So this is like a sympathetic consolidation or the refinement. So what the central nervous system is responsible for simple things like hearing, for example, hearing, language, touch, light, getting into your system, receptors, getting all this information and passing down through the series of synaptic connections. Movement is also the big one. So we either can inhibit or we can excite the postsynaptic neuron. So here, with the presynaptic neuron, we can release different types of neurotransmitters. So this could be a glutamate. It could be aspartate. It could be a glycine. Could be GABA. Could be serotonin. The thing is that glutamate, for example, is known for excitatory neurotransmitter. Aspartate, excitatory neurotransmitter. Glycine is inhibitory neurotransmitter. GABA is inhibitory neurotransmitter. Serotonin, it could be both. It could be excitatory or inhibitory. It depends where it's released. It could be dopamine as well. Can also be excitatory or inhibitory, and it can also be the nicotinic, cholinergic. By the way, cholinergic, it is acetylcholine, cholinergic. So also could be acetylcholine that also can be excitatory or inhibitory. So basically in presynaptic neuron, we can release either excitatory or inhibitory. It will bind to the receptors on the cell body, on the dendrites, somewhere in the axon helix, and transduct the information further down. So if here would be excitatory, any of the excitatory neurotransmitters, so the second neurotransmitter in the series will send action potential towards the next one, and so on. But if, for example, on the number 3, there's another neurotransmitter that is acting, and it's releasing inhibitory neurotransmitter, and there's, let's say, also another neurotransmitter acting on the 3rd one, and also releasing inhibitory neurotransmitter. So this neurotransmitter will inhibit or will stop progressing conduction down to next neuron. So what is inhibitory or excitatory? You remember this, right? In order to excite the cell, we need to depolarize the cell. Depolarization, you're bringing the sodium and you depolarize membrane. You go from, let's say, -70 to +30. So excitatory neurotransmitters, they depolarize. Inhibitory neurotransmitter, they're hyperpolarized. They remove potassium, because by removing potassium, you go from positive to negative. You go to hyperpolarization. This is a depolarization. You can watch the previous videos on depolarization, repolarization. So here, excitatory depolarized membrane, by bringing sodium in, so you depolarize. You can also bring calcium, right? You can open calcium channel and you depolarize. But hyperpolarize, you go like to -90, you go to the refractory period. You can remove potassium or you can bring negative calcium. You will definitely hyperpolarize membrane. And when you hyperpolarize, you inhibit that message that needs to go down in series. The thing is, when we're born, we already have all of this wiring. So through life, when we're progress through space and we are exposed to the external environment and we go through the learning process, we create more synapses. We do not develop any new neurons. So we create more plasticity. So there's more synapses on a particular organ. So like, let's say this is the organ, right? And here's a synapse. You can create collaterals from the main axon. You create more synapses. So that's a plasticity. Or it's called synaptic consolidation or synaptic refinement. Right? Hold the baseball this way, or hold the fork this way or this way. So the more you experience a certain task, the more synapses you create, the more consolidation in between these neurons and the refinements with the synapses. That's what it's called in all together, called plasticity. So one neuron can release one or few neurotransmitters, otherwise it would be gibberish. So if this one releases glycine, and glycine is a inhibitory, then it's supposed to inhibit this neuron, right? But then you can have another neuron, right, acting on this neuron, and this neuron can hypothetically release inhibitory. And if this neuron activated and it's releasing inhibitory neurotransmitters and this releasing excitatory neurotransmitters, and kind of cancel one another. But creating this consolidation or refinement and a particular task, if you're constantly inhibiting this neuron and this neuron also inhibits, right? Then when you excite this neuron and the two neurons inhibit this neuron, then obviously this message will be passed down. That's a general understanding on how the presynaptic and postsynaptic neurons work within the central nervous system, within the brain or the spinal cord. And you remember that everything works on this principle, plus, plus, negative, negative. So inside of the membrane is more negative, and outside of the membrane is more positive. By the membrane, I mean the axon or the cell body, or the dendrites. That's how we're able to conduct that electrical impulse. So by bringing sodium inside, you depolarize, you send down that electrical impulse, you conduct this transmission. And quickly, so the glutamate and aspartate are important neurotransmitters in learning and memory. GABA is important neurotransmitter in neuron firing in the brain. Serotonin, pain modulation. It could be excitatory or inhibitory. Dopamine is movement, motivation, reward. Acetylcholine, attention, arouser, memory as well. So these are the main central nervous system neurotransmitters. So this thing is called synaptogenesis. Synaptogenesis. Genesis, generation. Generating synapses in between neurons. Refinement. And within the brain there's a certain lobes that have a responsibility. So temporal is hearing, occipital is visual, right, hearing. Then parietal is touch, and the frontal is planning and problem solving. And all of this are here, and they constantly, each lobe communicate one with another. You either depolarize, excite, or you hyperpolarize. You are inhibit. Next in series. This was the third installment on central nervous system, on presynaptic and postsynaptic neurons.